everybody. Today we're going to be talking about DNA structure from the ideas of Watson and Crick. First, we're going to be talking about nucleotides, which are the building blocks of all DNA, and they are composed of a sugar, a nitrogen base, and a phosphate group. And each carbon on the sugar is numbered 1', 2', 3', 4', and 5'. 1' always starts where the nitrogen group is bound to the sugar. 2' either has a hydrogen if it's DNA, or an OH if it's RNA. This 3' binds the other phosphate group from another nucleotide, and then 5' houses our phosphate group. Now we have the structure of DNA here represented by some pasta, and if we look, the top group reads left to right of 3' to 5', and the bottom group reads 3' to 5', but from right to left, making this an anti-parallel structure. Now we're going to be talking about the nitrogen bases. Adenine, thymine, guanine, and cytosine. A and T bind together, and C and G bind together. But we see that A and G are purines, which is a one-ring structure, and T and C are pyridamines, which are a two-ring structure. So now we're going to be talking about bonds, backbones, and base stacking. Here we have A and T and G and C. They're each held together by hydrogen bonds, but A and T have 2 hydrogen bonds, and G and C have 3 hydrogen bonds, and that is a really big distinction between the two. Next, we have our backbone, and we have our nitrogen base pairs here in the middle, and on the outside, we have a phosphate backbone, and these help with the structure of the DNA. Next, we're going to talk about base stacking, and we have our nitrogen bases here, all connected by hydrogen bonds, but these bases are stacked on top of each other to give themselves a stronger structure because these hydrogen bonds are really weak, and if they're held together just by them, they would just fall apart. Now we're going to talk about handedness and grooves. If you look at this piece of pasta to the right and follow my pointer, it seems that this DNA is pointing up and to the right, making it right-handed. And if we look at this string, follow my pointer, go up and to the left, making this a left-handed DNA. Then from this string, we can see the minor and major grooves. With the major groove, this is a really big surface area, or at least compared to the DNA, and this is where a lot of the protein binding happens. Next, we're going to talk about stabilization. So we have this DNA molecule, and it has this phosphate backbone that has a negative charge. Well, if we take this DNA and put it in a high concentration salt buffer, the ions from the salt will interact with the negative charge of the phosphate backbone, making the backbone neutral. Well, what this also does is it increases the melting temperature. And if the melting temperature is increased, this creates a much more stable and strong DNA structure. And here are my references.